Hello everyone, welcome to sqadia.com. My name is Uzair Hashmi and today the topic of our discussion is cellular energetics. This chapter focuses on the discussion of the energy processes that occurs in the cell, and we are going to start off with the process of how energy is being produced in the form of ATP and what are the different processes that takes and plays an important function in the establishment of energy, that termed as the cellular energetics. This chapter would be focusing on primarily the discussion of the production and then the utilization of the proton force, or usually known as the proton motive force, that is specific for the hydrogen ion gradients. So we'll start off with the discussion of the glucose metabolism, and we are going to discuss the glycolytic pathway, and then we are going to discuss both the anaerobic and the aerobic metabolism of glucose, and we are going to see what are the different end products that we obtain in either in the absence or in the presence of oxygen, and we are going to see under what conditions lactic acid is being produced and under what conditions oxygen carbon dioxide is being produced from the glucose, and net result is the production of ATP as energy source. Now, moving on, we are going to discuss the fatty acid metabolism and the TCA cycle, also known as the citric acid cycle or the Krebs cycle. We are going to start off with the discussion of the aerobic oxidation of pyruvate and both the fatty acids. Now, it's important to consider that the metabolism of the glycolytic pathway produces a compound that is pyruvate, which undergoes several different addition, which ultimately is converted into acetyl-CoA, which then enters into the Krebs cycle, also known as the TCA or the citric acid cycle. Similarly, we are going to see what are the different degradation components of the fatty acids and how fatty acids are being converted into fatty acyl-CoA and then into acetyl-CoA, and how they are being responsible for the production of energy in the form of primarily NADH. Then we are going to move on towards the discussion of the citric acid cycle, TCA or the Krebs cycle, and then we are going to see how acetyl-CoA gets integrated into the Krebs cycle, forming from the oxalourethate, and then we are going to move on towards the discussion of how the citric acid cycle produces, that is primarily the NADH, two molecules of the NADH and one molecule of 5-adenine dinucleotide or FAD, along with the production of a GTP molecule as well. So this would be followed by the discussion of the Mallet shuttle, and then we are going to see how this plays an important function in the transport of the protons that an NADH contain, because NADH are not permeable to move. They are not permeable, that is, they cannot move from outer side of the matrix to the inner side. Then we are going to move towards the metabolism control, and we are going to discuss the fatty acid oxidation in mitochondria and peroxisome, and we are going to see what are the different what is basically the difference between the oxidation of the fatty acid that is occurring in the mitochondria, on contrary the oxidation that is occurring in the peroxisomes. So there is a little difference. And then we are going to see the allosteric control of the glucose metabolism, and how we are going to, in this case, we are going to discuss the role of insulin and how it basically initiates the production of the fructokinases that plays a key role in the metabolism control of the glucose. Then we are going to see what are the different changes that occurs in the redox potential and how that is being converted into the free energy and in the production, in which the electron basically move down the energy gradient, losing its energy, which is being taken up by the protons to move from one side to against the concentration gradient, so that these protons can derive the proton motive force in the production of the ATP from the ATP synthase, that we are going to study its structure in details as well. So moving on towards the generation of the proton motive force, we are going to discuss the multi-protein complex in the respiratory chain. We are going to see what are the different complexes that are present in the electron transport chain or the respiratory chain. We are going to see the plasto PQ-PC complex. We are going to see the Q complex as well, the cytochrome complex as well, and how they plays an important function in both the transport, the movement of the oxygen ions, and the proton motive force generation by the movement of the proton by against the concentration gradient. So moving on, we are going to discuss the cytochrome C and the Q cycle and their important functions in the generation of the proton motive force, and we are going to discuss the structure and the function of the ATP synthase. This would be followed by the continuation, of course, of the discussion of the structure and function of ATP synthase in harnessing the proton motive force, in which we are going to discuss the need of the F1 for the ATP synthesis. Now, F1 is a component, is one of the two components of the ATP synthase, where we are going to discuss the need for the F1 for the ATP synthesis. Right, this would be followed by the discussion of binding change mechanisms for the ATP synthesis. We are going to see how ATP synthase rotates in 3 conformations at 120 degree through a particular subunit that we are going to study in detail, resulting in the production of the ATP synthesis. And lastly, we are going to discuss the phosphate and ATP dependent transport systems, in which we are going to see the antiporters function in the transport of ATP and the phosphate group, so that they are going to produce the ATP. So I hope you're excited to learn more about this lecture. I'll see you in the coming sections of the video. Keep watching sqadia.com.